let's start with the enzyme lab. Does anybody just have anything to share about their experience about digging in and trying to get these rates and everything?
I have a question. As I was trying to figure out my slopes and stuff, my slopes like never leveled off my delta X over delta Y for each trial. Like I was expecting like 4 and 5 to kind of start leveling off, but they kept like, so when I did the final graph for all 6 trials, it just kept increasing. So I think I'm picking like the wrong area for like a best fit line.
So what then? So that's the.
That's like a whole different thing. I know.
No. I mean, why do you think you're picking? Why don't you just let your data be what it is?
Okay, because I feel like because we were talking about in class how like the final one would probably like end, and so
we said we're going to find out. We'll find out.
Okay, so that could be right.
Of course, it's whatever okay, cool. What would your conclusions be given your analysis?
So I would say that the substrate hasn't reached full concentration. I mean, saturation. So I just, whenever math's involved, I always kind of assume I'm doing it wrong. So that's why I was like, I don't know if this is right.
No, that's a perfectly viable conclusion. Is if you
okay.
if you feel good about getting your slopes,
yeah,
and you plot substrate concentration versus rate, and it just keeps going up, I would think you haven't hit saturation yet.
All right, cool, great, thank you. Yay.
Yes. And the thing I would also say is that there is a level of subjectivity in how you draw those lines. So 2 people might come to slightly different conclusions, and neither one would be wrong. They've just been kind of taken one path or another path where you can, is it this or is it that?
Okay, thank you.
Sure. Other questions about the analysis of this? Okay. So we're going to look at a few of just some examples that people have turned in, and talk about what looks good, what looks like maybe we need to, they should have, you might want to think about doing differently. So let's do that. Let's see. Share. Oh, there it is. Okay. And I have redacted everything, so it's all, yeah. All right, so let's see this person. So this person, the way they decided to do the graph, this, and like I said, this works just fine, is to put them all on one page. Although I would say if you're going to do it like this, this doesn't give you a lot of wiggle room to actually analyze things. Right? We know that everything interesting is in the first 60 seconds, but that 60 seconds is all scrunched in one little part of the page. Like this x-axis goes up to like 105, which, I would probably design my axis so it's more spread out. And also, if I was going to do something this busy, I would probably put this horizontally, right? So I could have my x-axis be along the long end of the page. Given that you are using these graphs for these analytical quantitative analyses, you definitely want to kind of open it up there. Another issue here is, this is always weird. Like, why are there 2 points for the exact same time? But if I look at these pink lines, this line that this person's adding for a slope here, it really doesn't feel, in fact, I look at this blue line, like where the graph is really rolling along, this line should be close to where my dots are. But the line is kind of like down below. So I would not say this line is probably a very good approximation of what the slope should be for these graphs here. So that's kind of an issue there. Let's see. Heck. And then let's go to the next page. How do I go to the next page? I see. There we go. Yeah, but their calculations look all right, right? So we take a look here, and they said for trial 1, they had 1 point. They said 50 was 15 and a half, 30 was 10 and a half. If we go over here at their lines at 50, actually, we don't. Yeah, wait, let me do that again. 15 and a half and 10. So I go here at 50, it's about 15 and a half. At 30, it's 10. So, they calculated the slopes well, but I think that this graph would make it tricky to get good slopes just because it's kind of crunched. And also just if I look at how they fit the line, it doesn't look like it's really going along when the reaction's really going nicely. All right. So, but then, then they do calculate, they got for everything here. And then they, made this graph, which is what we need to make. So that all looks good. So let's look at this person. So this person chose to draw each one of the graphs separately, which makes it easier to kind of see and to draw, although you don't get that nice comparison that you can see them all together on the same thing. However, we're going to run into some issues here. Whoops. Heck, goofy thing. Come on. Something, what happened? Hold on a sec. Okay. Why is this? I'm not used to using this way. So it's obviously, okay. My apologies for.
I think you can just click on the screen and drag.
Oh, here, it's down here. I can. Okay, there we go. All right, so what we have here is, first of all, when you do the calculations, you do want to actually make it clear where you're getting your numbers from. Like the last person did that nice little, those perpendicular lines for rise over run. This person did not do that. But still, if you read and you look at their numbers, you can figure out what they claimed they were doing. So I'm going to do this marquee zoom. Here we go. So and they're saying like when they were at 30, right, let me do the annotate thing here. They said at 30, it was 10 and a half. So I go to 30, and that's about 10 and a half. And they said at 16, at 15, it was 6.3. I go to 15. Oh, no, so I should, it gets a little tricky. This is 7.5 here. So it gets a little tricky, but that's okay. All right, so now I take it. Okay, so this actually, so then they calculate this, they get their number. Well, shoot, wait, I just spazzed on you. This one, if you take a look here, this is kind of similar to what I was talking about with that last one, right? If you take a look at this line, it's really not going along when the thing is really rolling along. It's totally like kind of at a weird like teeter totter, right? The top is leaning down towards where it's dead. The bottom is leaning up away from where it's really rolling. Like if I was going to draw a best fit line for this thing, I would want a line. And last time I learned I can use lines. I would want a line, that's kind of going like where the dots are really going, when it's really rolling along, right? This line here is not giving me a good, well, I need to, who does it? Trying to, so can you see that? We have somebody who's kind of loud. I think, Dean, can you mute? Okay, thank you. So do you see that? Like this line that they drew really is also kind of not where it's really rolling. So you want to think about your drawing your best fit line when you're doing this. And it would be nice to have, if you were getting it from 35 to 20, then you should go from show me that so then I can really see where you are getting it on the graph there. 20. Yeah, so anyway, so just putting that out there. I need to stop the, let me clear this. And okay. So that looks good. There it really does seem to be going along where, and here they kind of corrected themselves. They realized this is really off. But even this one is still, I would say, could be a little, if you really wanted to kind of get where it's really rolling. So anyway, that is that one. Whoops. This one. So this person actually used, they kind of printed out, which is fine as well. I think it's, easier, nicer. And again, on this, there is no reason to print out past 70 even, but it doesn't. But here, like when they drew their line, it would have been nice if they drew it longer to where, because it should be fitting from 10 seconds on. So if I drew this, I would probably want my line longer, so I would really get a more holistic sense of how does it fit into the beginning of the reaction here. Again, for the most part, this all looks really nice, but there's no reason to keep the line so short. But then and here, this is what I was talking about, having the nice little lines showing where you actually got the numbers to do your calculations. Oh, and this I should also mention. So this is good. What they did on trial 6, they know that, the first few seconds were kind of a mess. If we look back, so they only took the slope of when the reaction's going, which is when actually it's not going because the reaction's not going. If we look at this person, we look at their trial. Oh, yikes! We look at their trial 6. We got this weirdness going on here. Trial 6, they actually calculate a rate. Whoops. Dag nabbit. Sorry about that. There it is. They calculate this rate here, and it doesn't make any sense that there's a rate there, right? Because there's no substrate. And then on your graph, you'd have some big explaining to do if you calculate the rate for this. And let me get it up. What did they find it? They ended up with 0.5 mils of oxygen per second. That would mean on this graph at 0, this should be up at 0.5. So this whole graph would look really weird. You'd have some big explaining to do if that was actually true. So that data is actually not on this graph because 0 should be 0.5 because that's what they calculated if they were being consistent. So be careful about how you treat trial 6. It is data. It's 0% of the substrate, and there is a slope, but it's 0. If it wasn't 0, something would be really wrong in our experiment. So hopefully, does this help? Does this help to see this?
Yes.
Okay, and again, I'm not doing this to make any of the people, the 3 you saw are the 3 that people turned in. Each one got turned in with its own challenges, and the fact that people actually are on top of things to get things turned in this quick is impressive. And at the same time, this lab in particular is one that people always have trouble with. So it's worth spending a little extra time and then take into account what I've just been talking about in terms of thinking about calculating your slopes and both the mechanics of drawing your graphs and the more subjective thing of how to place that line. And again, the line is subjective. So you could end up having 2 people analyze the same data in a responsible fashion and still get different results. So that happens. All right, any questions about Enzyme Lab before we walk away from it?